Okay, so I'd like to welcome everyone to the Plant Response and Adaptation to Abiotic Stress session. We have, I think, four world-class speakers, and their talks are going to cover, most of the focus is on soil-related abiotic stresses relating to mineral nutrition and water, but we have a wide range of, or a range of different talks involving long-distance signaling and that role in abiotic stress tolerance, vascular biology, breeding for better roots. So it's going to be a very interesting session. So it's my pleasure to introduce the first speaker. It should be well known to the audience. I've known him for over 40 years, Professor William Lucas, who's a distinguished research professor at the University of California at Davis, where he's been a professor of plant biology for over 40 years and was chair of the Department of Plant Biology for the last 10. Bill's work has really he made seminal impacts in two research areas. Early in his career, it was a world leader in plant membrane transport, membrane biophysics. And then about 30 years ago, about the time I left his lab to go to Cornell, he moved into a new area related to transport, but the transport of long-distance transport of information and signaling molecules in plants. And his work led this new field showing that the phloem is an information superhighway, chock-a-block with different RNA signaling molecules and proteins. And he's going to be talking about some of that today. So I'd welcome Bill up here. Oh, wait, before that, certainly a very accomplished scientist with lots of awards. I just want to mention a couple of them. He has received the American Society of Plant Biology Gibbs Award. He's a foreign member of the French National Academy of Sciences. He was one of the first group of highly cited researchers in the plant sciences. As his recognitions go on and on. So I'd like to welcome Bill up here to give his talk.
What I want to do this afternoon is give you a little journey. The role of the vascular-mediated long-distance signaling in crop improvement. I want in this time to try to show you that there are, in fact, so many signals. As Leon said, it's an information superhighway, and in the time allotted, I'm going to give you just a few discrete examples of what we know, where we are, and where we're going. So as you can see from this the slide, these are the attributes or the traits that we already know for at least 20 to almost 50 years, in some cases, of the traits that are controlled by the vascular system. Now, I'll apologize if you came to listen to me talking about the xylem. I don't have time to talk about that today. I'm going to focus on the phloem, although we have very interesting information on the xylem. So as you can see from the top, flowering time, so this is very important for yield, plant architecture, for root shoot ratio, for branching. All of these are very important for crop production. Mineral nutrient signaling, the optimization of plant growth. We heard something about that with Giles. The allocation of fixed carbon, control over yield, very, very important. We know the system is doing it. Right now, generally, we know very little about it. And then systemic acquired resistance in terms of resistance against a wide range of pathogens, so yield protection. And then very recently, not going back 20, 30 years, but systemic gene silencing, so the control over RNA stability, and recently the control over genome stability, which was hinted at for some years, but now we really have direct evidence. So the question that I'm going to present to you is, so how does the phloem control these traits? We need to understand. We, in our lab, pioneered the development, not of Arabidopsis as a model plant, but the cucurbits. And it is the reason is because, as you can see here, the phloem sap from a stem, an excised stem. You can see it on the other side too. So we can collect phloem exudate from so CSR on the left-hand side. CSR is the central source region. And we have a cucumber genome, an annotated genome, and we have, in fact, many other accessions that have been resequenced. So we've got considerable resources. So then the question that I'm going to present to you firstly is, what's in that phloem sap that you could see? So up there you can see the phloem sap. We collect that phloem sap, and then using the genome and mass spectroscopy, you can see from this that we can develop a phloem proteome. And as I put on this slide, the phloem exudate contains about 2,300 proteins. I mean, this is amazing. When I was a student teaching people like Leon and Davis, we used to tell the students that the phloem carried sugars, amino acids, and some hormones. Now we are discovering that there's a huge number, and I'm going to show you material that I still, right now, I don't understand. But here's what we can see. So when you look at these proteins, there are, as I say here, there are many surprises. And so what are those surprises? Well, for example, proteins with RNA binding function here. This is very interesting. Well, RNA binding. But, I'm sure you all know, because you've passed your qualifying exams, that the sieve tube system, when it's functional, doesn't have a nucleus. So why do we have RNA binding proteins there? Well, I'll show you some more about that in a minute. Not only that, look down here. So we have cell cycle, cell fate, and developmental regulators, transcription factors. There's a lot of transcription factors, and I'm going to come back to that in a minute. Okay. Back over here. Okay. So the phloem delivery of proteins involved in plant architecture. So Leon was mentioning about root architecture. So we're going to look at what is in the phloem that's controlling root architecture. So first of all, I'm going to give you a brief journey down the river of the phloem in terms of root-to-shoot ratio. And then I'm going to talk about the control of this process, not only in root architecture, but in terms of nutrient acquisition, specifically nitrogen. So for a long time, we have been measuring identifying proteins in the phloem, and one protein that's been identified in a range of phloem exudates, not only cucurbit, but other species, is Digeotropica, which is a mutant that has been known for about more than 70 years. And it's an auxin-insensitive mutant. And as you will see, I hope, so here is the Digeotropica. It doesn't have any lateral roots because it's auxin-insensitive, and it only has these adventitious roots. Now, if you look at the panel on the right, far right, this is wild type. If you look at a cross-section here in the stem, you can see these large metaxylem vessels. They're absent in this Digeotropica. Now, I love doing grafting. It's a very powerful experimental tool. So when we now graft the Digeotropica onto, wild type onto DGT roots, you can see we transfer them to this. That's the same as wild type. When we now look, you can see these large secondary metaxylem vessels have been restored. You go down to the root, you see the same thing. They don't make lateral, so this is no lateral roots. This is lateral roots and large metaxylem vessels come down here to the mutant, and now you've seen it completely restored. So this is a very simple assay. So that means, and that's a single gene mutation, Digeotropica. So what does this mean? What I show you here is, again, when we use grafting. So now we can look, and this is in tomato, of course. Previously it was tomato. So you can see the protein in the stem tissue. And this is in Digeotropica, no protein. And now this is the grafting in the stem. You can see the protein, and you go down to the root, and you see the same, that the protein is coming from the shoot through the phloem into the root. Again, you can see it here in terms of Digeotropica, and we have an antibody for the protein. And you can see here in the control, you can see the protein here in the phloem. This is the xylem back in here. Now you can do the grafting and compare this. You can see beautifully the signal in the functional phloem. And this is the control. This shows you in a transgenic tomato line that it's not the RNA. So in this case, it's protein moving. What does it do? So now let's look at what happens. So this is the grafting. This is now with a reporter for auxin. So you can see DGT, no auxin. This is wild type over here. And so if we follow this in the confocal, you can see that the lateral root emerges. This is all normal, that's wild type. Now come down here. Now we have this reporter for auxin in the DGT, and we're going to do the mutation, the grafting. And now you can see it's exactly the same. We've completely restored the ability to make lateral roots. That's a single gene mutation that's responsible for something moving in the phloem. It itself is moving in the phloem, so it's a chaperone, so it's most likely carrying a signal. Now, what happens in terms of transcriptomics? It's quite remarkable. If you look at this, this is the most important section, so you can see that heterografting of VFN8, the scion, onto the GGT rootstock restores expression of these about 2,600 genes involved in root development and function, specifically in root development and function. And when you look at the map, now you can see the rescued gene set, the rescued transcription factors, and more importantly, the rescued NAC domain transcription factors. And this is remarkable. Look at this, okay? So now you can see DGT, okay? All of this is upregulated. You can see the red. Come over on the other side to the wild type, blue. Now you do the grafting, and you can see the restoration down the line, except for this one. This is a very important NAC that's involved in controlling root development, and so you can see that it is downregulated, and then when you graft it, it's restored. It goes the other way. So what does this mean? So now we have a signal coming down, so cyclophilin is coming down through the phloem, and it's acting in some way to restore this process. So we asked the question, does this report something about the physiological state? Because my lab has always been interested in what's controlling root-to-shoot ratio. So we went to look at that in detail, and as you can see on the left-hand side, so the trafficking of this cyclophilin is normalized to the trafficking of GFP, and you can see at the different light intensities from 150 up to 600, you can see that the amount of cyclophilin that's trafficking is controlled by the light intensity. Now when you go down and look at the bottom panel to the right, now you're looking at the shoot-to-root ratio. You see DDG is insensitive to the light intensity because the color code is the same as the intensity in the top left. And so now when you do VFN8, the control, you see very nicely that as you increase the light intensity, the shoot-to-root ratio decreases because you've got a need for, as the light goes up, you need more water and for transpiration, for photosynthesis. VFN8 over DGT, you see you've restored that capacity relative to the DGT. So what does this tell us? So the model that we have is that cyclophilin functions as a regulator to control root-to-shoot ratio. So when you look at this diagram, you can see that the photosynthetic capacity is increasing with light intensity, so does the expression of cyclophilin. The level in the phloem goes up, comes down, and augments the level of cyclophilin that's in the root to drive this process of regulating lateral root development, increasing the ability for ion and water transport, coming back to give rise to supporting higher photosynthesis. So that's one example. Now, I want to give you another example, and this is a transcription factor. So if we look at the Arabidopsis experiments with high 5, this is a phloem mobile transcription factor as shown by these data from Chen et al. In Current Biology. So here's the mutant, the high 5 mutant, and the signal. So you can see shoot and root, it's 0. This is now in this transgenic plant, in the Hi5 background. So you see the transcripts are in the chute and barely detectable in this system in the root. Okay, so now, and there's the Western blot. So you can see that although the transcripts aren't there, the protein is there. Now you come down to this nice grafting study again. And when you look at this, you see Hi5, there's no GFP signal, which means that there's no GFP signal. Now, when you look at this with the promoter driving this, and you graft it onto the Hi5, you see signal. Now, keep that in your mind, because it's going to be 2 slides later, you're going to have to remember it. It's difficult for you, Leon. Try hard. Okay, so this is a beautiful picture. Okay, so this is the one that you just saw, so that you can remember it. This is Hi5 over here as the scion, that is the shoot tissue, grafted on to this guy, which is expressing in the root, and you see no signal. Okay? This is remarkable because here's the gene. But I don't have time to show you that Hi5 binds to its own promoter element. So if there's no protein coming down, there's nothing to start the process. Look at this guy. So that's the same, except it's not Hi5 here. Okay? This is not the mutant here, it's just the transgenic in the mutant line, and look, we've got fantastic expression. So what does that mean? Why is it doing that? Well, I think you can see it from this slide. So, if you look at, this is wild type, so this is now nitrate transport into the root. Okay, so you can, and this is to remind you what happens when you have a Hi5 coming down through the phloem and egressing beyond the phloem and activating expression in the root tissue. So now you can see in the first line that you have about 25 units of nitrate uptake. That's the control. So that's that's running in the control. So the next one over is the one that we showed you. So go to the third one. That's the case where it only comes down through the phloem, and there's no Hi5 gene with the promoter in the root, and so now you don't have this autoregulation. So in a nutshell, this Hi5, its level is adjusted by light intensity, and you can see the long-term effect in terms of growth here is that under normal conditions, there's a balance between carbon and nitrogen, but in the absence of this signal, now this balance becomes out of balance. This is a very pretty story. So this is a transcription factor. So the general scheme is very similar to what I showed with cyclophilin. So now you have it, the promoter, you drive it, it comes down, it augments what's in the root and drives further production depending on what is coming down and upregulating it to control nitrate transport. And nitrate, of course, the availability also drives lateral root development. Okay, so now let's go on to phloem delivery of mRNAs involved in nutrient signaling. So I've talked about 2 proteins. Now this is the work of John Zhong and Brian Ham, who's sitting down here in the audience and who's recently joined GIFS and the University of Saskatchewan. So what John and Brian were able to show is that when we're looking at phosphate stress, and this is now a cucumber, we stay with cucumber now, not Arabidopsis. So when we look at the central source region, when we look at the central source region, 12 hours after a phosphate stress in the root system, look at this. The phloem sap changes the mRNA levels in an amazing way. Roots and apex, etc., there's hardly any change. If you look at the heat map, you can see right here in the phloem, huge changes. Lots and lots of RNA molecules being upregulated. Now, this one's one ahead of me. Let's look at 24 hours, but now not in the central source region, but we're going to look up in here, in the developing leaf and the shoot apex. You can see the distance is about maybe 5 to 6 millimeters. The phloem is coming up from the source region and it's delivering molecules, and then of course going down to the root. That's a lot longer distance. But look at this map. I mean, this is remarkable. This is under phosphate sufficient conditions. These are the unique genes that are in the developing leaf, the shoot apex, and the root. When we change it to phosphate stress, this population doesn't change, although they're unique. But look at these are turned off, these are turned on. A massive change, and these are coming from the phloem. These are, as I show you here, this is cucumber, this is watermelon. So we can identify cucumber RNA in the watermelon developing leaf and shoot apex. There's a massive information superhighway here of RNA moving. So when we look at these genes, as I say in the heading, these are characteristic mRNAs characteristic for proteins and responses that have been shown in other systems, including Arabidopsis, that are representing the phosphate stress syndrome. So here we have an amazing amount of RNA moving, and specifically moving to individual locations on the plant. Developing leaf and 5-6 millimeters difference, you've got the shoot apex. And yet the population is entirely different. So this tells us that the phloem, and I don't have time to talk about the xylem. We have data on the xylem, but what we're talking about here is that the phloem, an entirely different new population goes up to the shoot, a different population comes down to the root to reprogram the genetic program for phosphate stress. And this is the vascular signaling. And this is specific. We can get this material specifically. If you go and look at what other people have done, they take the whole plant, or they take the root and the shoot, but they don't put the two together. This brings them together. So, now, Brian Ham, before he left my lab, there was a very, now Leon will know, I've always been interested in NPK, right? I mean, if you're in an agricultural college, it's all NPK, and we heard a lot about N in Giles's talk. So if we can do this on phosphate, well, let's go do it for for RNA, let's go and do it for phosphate, nitrogen, and potassium for the proteome. So we can make a proteome, we can collect the phloem sap from these different locations, nearly did it, from the apex, from the central source region, from the root-shoot junction, which is telling you what's going down to the root. We can do the mass spec, so we can quantitate it using iTrack labeling. And from this, we get another big surprise. So what Brian was able to show with the colleagues working on this project was that we have many changes in the nutrient that are nutrient-specific. So you can see here, for nitrogen, this many proteins are changed specifically. If you look at phosphate, this number. Now this, I have to tell you, this is two peptides in the G1 and the G3. If we look at 3 peptides, then we have about I can't read it from there, but I can read it from here. Something about 40%, and this one is about 70%. So that's of the total 2,300 proteins. So at least conservatively, 50% of the proteins that are produced in the central source region are moving. And we're in the process of identifying which ones are going up and which ones are going down, but we haven't got that done yet. So, I mean, honestly, when we got these results, I find it almost enough to keep me awake at night. What's it telling us? I don't know yet. So I want to tell you about one of those proteins that is in the phloem that changes under phosphate stress. And this is the work of Jae-Yu Chen in the lab. And shortly she will be coming up here to GIFS as well. Everybody's stealing my lab. They're coming to GIFS. So what I'm going to show you very briefly, because of the time, the characterization of this phloem phosphate stress repressed 1 protein. Interesting protein. So there's downregulation of this PPSR1 in the central source region. So you can see that from this top here, this is within 24 hours, and if you go up to the top of the plant or down to the base of the plant to the root, they don't change. You can see it's still the same. They change later, and I'll show you why. But this is a very quick change in this protein. It's dropping down, and you can see down here on the phloem sap, after 3 days and 7 days, it's gone. There's nothing there. Okay. So, how is that possible? What's the mechanism that can cause this to change? Well, a number of years ago, we reported that the phloem, you remember it's E-nucleate, and it has a few organelles left. It has parietal endoplasmic reticulum, and it has mitochondria and plastids that are anchored to the side. They have 26S proteasome. They have a lot of ubiquitin. They've got the enzymes for ubiquitination. So Jayu thought, maybe it's somehow being turned over through the proteasome. Okay, the 26S proteasome. So, okay, so she tested that. And so you can take this protein that's tagged, so we can purify it, and we can add it to phloem sap that's collected from the shoot apex. So you see all of this is shoot apex, and this is the central source region. You notice here that there's no degradation even after 24 hours. And the test in here is that we use MG132, the 26S proteasome inhibitor. So you can see in the presence or absence of that inhibitor, no change. Very different down here. So now you can see that only the phloem sap collected from the central source region has the capacity to degrade this protein in this assay. So Jayu thought, well, what would be the control to tell the protein to be degraded? There's got to be some signaling process. So what might it be? And so Brian Ham had done an experiment earlier asking about phosphorylation and dephosphorylation. So Jayu thought, well, it's worth a try. If Brian can do it, so can I. And so she did this, and it was a very nice result. So as you'll see from this panel, left-hand side panel, it's highlighted in red, that this is NGL phosphorylation, and you'll see there's the phosphorylation. All this is other proteins being phosphorylated. Now you can see it's not, in the case of minus phosphate, there's no protein. So she cuts this out, does mass spectroscopy very quickly, identifies the gene, and then works on the gene. Okay, so now what is it? And of course, it's a kinase, so it's PPSRK. Why wouldn't you name it that? Of course. So this is what happens, at least in the model. So you can see here that this kinase is unstable in phloem sap from phosphate-stressed plants. And that's a very nice example shown here. This is GST-tagged and purified protein. Now you can add it to the phloem sap, and you see it's degrading. When you use MG132, there's no degradation. So the model that she's developed is that this protein is phosphorylated, and we know it's we know it's phosphorylated, but we just don't know the site. So that phosphorylation occurs when you have minus phosphate. The kinase is turned over; it's gone, so it can't phosphorylate it. So now this region of the protein can be ubiquitinated, and when it's ubiquitinated, of course, the 26S proteasome is going to turn it over. So that's another nice example of, this case, a protein. And this is the model. So this model is being tested. So basically, under control conditions of adequate phosphate, this system is phosphorylated by PPSRK, and so now it can move to the shoot where it's enhancing growth. The model is that it goes in both directions, and we don't have the evidence of that yet. That's what I told you. Now we're identifying what is going up and what is going down, but I'm reasonably confident that it's going in both directions, and it's reducing root growth. The mechanism we can't talk about yet. Under minus phosphate, now this process, the kinase is degraded. Now we degrade, but through the ubiquitination process, PPSR1. Now that represses that relieves this process up here. So now we are blocking growth. Now down here, this repressin is relieved, so we have enhanced growth. So now we know under minus phosphate, we're going to shift from shoot growth to root growth. So that's the basis for the model. Okay. So I come back to the original. Now we know that we have up to as many as more than 2,300 plus proteins. We have several thousand mRNA. I didn't give you the numbers for that, but so many thousands of mRNA that are moving to specific sites. We have lots of proteins that can do many different functions. We have FT, flowering locus T, which we showed back in 2009, is moving through the phloem. Okay. We know other proteins that are moving to control tuberization, etc. So what I would say is, in the next generation, these kinds of these traits that we can describe here are going to be characterized, and I would be quite sure that there will be multiple signals that are controlling similar traits for redundancy. So high 5 and cyclophilin and whatever cyclophilin is carrying, they're doing basically the same thing, except that high 5 is actually going and targeting also to the promoter of the nitrate transporter to regulate nitrate transport in balance with carbon fixation and allocation. So I think we are going to begin to see an amazing complex array of proteins, RNA, and peptides that are moving through the phloem in a way that might be even more sophisticated than the blood going through Leon Koshin's veins. It's possible. So time will tell. Okay, so the acknowledgements. My lab, Brian and Jayu. My collaborators, Leon Koshin, of course, now Brian Ham is up here. Faye in Boyce Thompson Institute is a fantastic collaborator. Zhao Liang Zhong, or John Zhong as we call him, is now back in China. He was fantastic. San Wen Wang, I mean, without San Wen, a lot of this wouldn't be possible. He's the director of the Agricultural Genomics Institute in Shenzhen. Hong Liao in Fujian Agricultural Forest University in the first field trial we did on phosphate stress signaling. And Etvan Chen from Academia Seneca, his lab is doing all the proteomics that we talked about. Shmulik Wolf, postdoc in my lab 35 years ago, we still work together. We did the cyclophilin project together. And Chungming Liu, now at the Institute of Crop Sciences, we're doing an interesting project on the development of the phloem in the root. So we want to get that system worked out so we can see what's coming down to work at that site. And on that note, my last slide, I hope that I've convinced you that the phloem is a conduit for controlling many agriculturally important traits, and the actual genes and the proteins and the signals are beginning to be uncovered. I spent a wonderful 2 years in University of Toronto back in 1975. I'm so pleased to be back. My homeland of Australia, my present land, although with Donald Trump, if he gets any worse, I think I'll come back to Canada. But I'm in California, and of course, come to visit anytime in the spring, because these are the California poppies, and as Leon knows, north of where I live, there are huge fields, or as George W. Bush would say, huge, right? Huge fields of beautiful flowers, and we can go and either sit in there, or we can go and lie in that field and think about long-distance signaling or other things. Thank you very much for your attention.